Welcome, sir. Varman, sir. Sir, are you connected?
Hello, good evening, madam.
Good evening, sir.
Yeah, good evening. Okay, madam.
Okay. Okay, sir.
Sir,
Five,
can
within
you
5 minutes,
get
we'll start.
option for sharing, screen sharing?
Given,
Screen
sir,
share.
you could be able to share now, sir. Please check, sir.
Okay. Sir, one of my student also joined. Please let him, sir.
Could you please let me know the name, sir?
Vijay Raj.
Sure, sir. We'll give.
Okay, okay. Thank you, sir. Thank you. Is my screen visible, sir?
Yes, sir. Perfectly visible.
Shall I go to?
Yeah, slideshow, sir.
Yeah. Is it visible clearly now?
But not.
Okay, sir.
It's not in full screen.
Sir, could you please put it on full screen, sir, in slideshow?
Sir, hello.
Sir, please, sir. Tell me, sir.
Yeah, yeah. Is it now in full screen mode, sir?
No, sir. Still in the normal mode, sir.
No, I changed it to full screen mode. I don't know why.
Better you make it a slideshow and again try to share the screen, sir. And we couldn't able to see your student in the participants list, sir.
Yeah, yeah. He will
Waiting
join.
for
Sir.
him.
He will join. He will join.
Sure, sure, sure. Soon after he joins, we'll give him co-host. Sir.
Okay. Okay.
Right, sir.
Now I changed it to full screen, sir. Is it okay now? Hello, sir. I am having.
Sir, waiting, sir. Still,
You?
my screen.
Okay. Okay. Resume sharing. Sir, should I?
Sir, I couldn't see the content, sir. It is full screen, but contents of the PPT is not visible.
Now, is it moving or no?
Yeah, I could see the mouse pointer, sir.
Now, sir?
PPT is visible, sir, but not in present full screen
But
mode.
when I
Right?
go full screen mode, it stops, sir.
Now you try to give full. Press F5, sir. F5 on the keyboard, please.
Yeah. Just I'll try. Yeah, I pressed, sir. Now
No,
my
no,
screen
no.
is in full screen
No,
mode.
it's not working. So better, sir. Better. What we do is you go on full screen, then share screen from Zoom,
Share
sir.
the content, right?
Yes. You share the slideshow from Zoom, sir.
Slideshow from Zoom.
You can now stop sharing your screen.
Okay.
Switch back to your PowerPoint presentation.
Okay.
Make it full screen, then again switch to Zoom console, sir.
I could not see the Zoom icon.
You can press Alt tab, sir, on your
Alt, sorry.
tab.
Alt tab.
Yes, sir. Yes, sir.
Alt.
So you could able to switch through the windows.
No, it is not going.
Try Windows tab, sir. Otherwise,
Yes.
so now from Zoom you can click on share screen, and you can select the slideshow. The one which has slideshow, you can select that window, sir. Yes, perfect, sir. Perfect. We could see that India map. Some map is there.
Now is it okay?
Perfect, sir. Perfect, sir.
Okay, sir. Thank you, sir.
Thank you so much, sir.
Welcome, sir. Welcome. Shall we start, sir? Or?
Just wait for a minute or two, sir. We'll get the instruction from the host, then we'll go with the formal proceedings.
Thank you.
Right, sir. Thank you, sir.
Dr. Koslinda, we can start the session.
Yes. Pardon me, ma'am. I couldn't hear you.
We can start the session, sir.
Yes, ma'am. Yes, ma'am. Waiting for new staff, so we'll start.
Sir, shall we start, sir?
Yeah.
Yes, ma'am. Please go ahead, ma'am. Please.
Yeah. Good evening, everyone. Welcome to Atal Academy sponsored 2-week faculty development program, IoT-driven wearable devices for healthcare and defense application. Today, the session 4 about the molecular sensing for disease diagnosis. The resource person is Dr. Venkataraman Dharman, sir, Assistant Professor, Molecular Electronics Lab, Alagappa University, Karaikudi. Now, let's upon Ms. Chandana Kala ma'am, Assistant Professor, SciTech, give the introduction about our resource person. Welcome, ma'am.
Madam, I could not hear your voice.
My voice is audible, sir.
Yeah, your voice is audible now.
Okay, sir. Thank you, sir. Good evening, one and all. I'm Chandana Kala, Assistant Professor, Department of PCE SciTech. I would like to introduce today our chief guest, Dr. V. Dharuman, Assistant Professor, Department of Bioelectronics and Biosensors from Alagappa University, Karaikudi. He completed his BSc, MSc, PhD from University of Madras. He had 9 years of teaching experience and 15 years of research experience in various research institutes. Under his guidance, 4 members completed PhD and 2 members completed for MPhil. 1 project fellowship is going on, and he guided 21 projects for PG students. He published various journals and attended 100+ conferences. He received various funding projects under the agency of UGC, DST, CSIR, etc. He achieved 13 distinctive awards. He visited so many countries, that is South Korea, Japan, and Germany for technical visits. He is having life membership in different professional and academic bodies. Now, I'm very happy to welcome you, such a great personality. Thank you, sir. Welcome you, sir. Over to Nisha.
Thank you, madam.
Once again, welcome you, sir, for the today's session. Please carry on your session, sir. Thank you for your time.
Madam, just a moment. I reconnect my network. It is little bit disturbing.
Yeah, okay, sir.
Just a few moments, few minutes.
Yeah.
Just. Hello.
Yeah, sir.
Good evening to one and all. Am I audible now?
Yeah, sir. You are audible, sir.
Okay, madam. Thank you, madam, for the nice introduction. Sorry for the network disturbance at the start of the session. As the program is based on the IoT application for sensor application or wearable sensors, you know that wearable sensors are the one which are advanced or integrated with the internet. It is connected with the integration of internet, which is on the sensors, which senses the signal and it transforms the signal to the further process. So here, although it is a wearable sensor, basically the sensing principle and the mechanism of sensing is basically same for all the sensor devices. So what I thought was it is better to talk about molecular sensing for disease diagnosis, because all the diseases are based on the misbehavior of molecules in a physiological condition that leads to the inception of disease in our body. So, madam, may I request, is there any problem in my audio? Please inform me, because I keep on going. So, if you don't mind, please interrupt me. Okay, madam?
Definitely,
Yeah, sir.
sir.
Okay, sir.
Okay. So here, what I try to introduce is I'm from south corner of India, that is the from Tamil Nadu, and I think you know that because you are my neighboring state, you know that where from I am. It is south corner of Tamil Nadu, Karaikudi, and this is the block in which my department is located in Alagappa University. So here, if you look at the diagnostic application for the fields where you can apply, one is the food industries, and another one is the environmental monitoring, home diagnostics, and of course, point of care. That is where your IoT, wearable devices are fall into. And defense, and of course, research labs, all research labs are trying to develop some sensor devices for various applications, depends on their need. So if you look at food and food industry, where the quality of the food quality and the origin of food and the degree of contamination, these are the essential parameters to be monitored. And of course, in environmental, we most care about the air and water quality, and other things are like pesticides and biological oxygen demand. These are the most important application where biosensors find utility. And another important aspect is home diagnostic. This is where we are going to talk about how the molecules can be used for diagnosing various diseases. So here, the sensor is a device which can sense for a sense. Generally, we call it as the electronic device, which is able to sense the presence of any physical or chemical quantity. If it senses a physical quantity, we call it as the physical sensor. Otherwise, it is a chemical or biological molecule. We named it as a chemical sensor or biological molecule. Basically, it is a multidisciplinary subject, and it is the convergent of electrical, biology, chemistry, physics, material science. Why not the computer science also be joined in the development of biosensor devices? But recently, the IoT, that is Internet of Things, taken over the advancement of biosensor, bioelectronics and sensor devices to monitor the patient or on-field application of the disease diagnosis. So here, if you look at the class of diseases, generally we classify under the communicable and non-communicable. All are depends on the molecules which induces the diseases. For example, viral and bacterial molecules induces different types of bacteria and viral diseases, and other non-communicable diseases mainly classified as diabetic, neurological, and genetic based. So here, sensor by definition, it is an input device which provides an output signal with respect to specific physical quantity. That means if a person walk across a automatic sensor, your doors will be open, something like that, and it converts the signal from one energy domain to the another energy domain, either electrical or optical. For basic classification, passive and active, those things we don't need. So if you look at the molecular sensing, these are the devices. Madam, I will call you later, please. In these molecular devices, we mainly focus on sensing of molecules related to neurological diseases or some kind of cell malfunctioning or inducing molecules which goes across the cell membrane to take part in the chemical reaction where DNA is formed and which results in the formation of protein. If these process or somehow it is stopped, means then we get a kind of diseases. So here, why we need to have molecular sensors? Because all the diseases are giving some symptoms, so that based on the symptoms, doctors can identify or diagnose what type of disease or what type of infections that we get. But we cannot see the disease at the early stage, which is an important criteria for preventing the mortality rate. So that people are trying to look at the molecular behavior. For example, if you have a neuro-related problem, then there are certain type of chemicals which is which are responsible for inducing neuro diseases, or if you have a kind of DNA, kind of a cancer or other type of diseases which are related to DNA malfunctioning, then that will be indicated by change in the base pairs of DNA molecule which is present in the cell. In other words, the cell morphological changes, pathological monitoring, or you can also think about the ions which are transport across the cell membrane, where sodium and potassium are responsible for maintaining the cell potential at the rest condition. If there is a change in the cell potential, means there is a change in the ionic concentration across the cell membrane, that leads to that indicates the problem of disease related to kidney or heart kind of blood pressure, all those things. So here, why we have to look at is, if you look at the cell membrane, and each cell membrane consists of various receptor molecules, and these receptor molecules are going to attract the chemicals which are present in the bloodstream, and it transport across the membrane for further reaction to generate or to complete the biochemical reaction which is required for the normal function of the healthy person. If it affects, then we have to look at what is happening at the receptor stage, or which kind of molecule is able or not able to penetrate the cell membrane like that. So here the goal is, because this process occurs at a very significant rate, and the concentration of the molecules which are approaching the cell membrane are very, very, very minimum. Indeed, it is below femtomolar or nanomolar or micromolar, even below automolar, like that. There are concentrations of chemicals which are present in blood or urine. If you diagnose, if you are able to estimate or quantify the chemicals which are present in a physiological fluid, then you can correlate the disease state of the person. So for that, our goal is to maximize the signal, because why we need to maximize the signal means, here the quantity of signal which is generated at this surface or at this junction is not enough to be sensed by the sensor devices, so that you need to develop more sensitive devices. So for that, you have to enhance the signal parameter, which is being measured by using our sensor devices which are going to fabricate. So if you look at conventionally, biosensor is again analytical device used to identify or quantify the chemical or biochemical target in a given solution or body biofluid selectively or sensitively. That means it is a device which can detect chemical or biochemical target in a given solution selectively and sensitively. The well-known biosensor devices are glucometer and of course the pregnancy test. These two devices are well-known and cheaply available at the market. And these two, one is based on the affinity interaction between two molecules, whereas this one is by the catalytic interaction. So so by knowing the nature of a reaction that is happening in our body, then we try to develop the sensor devices for particular application. So if you look at the components of the biosensor device, it is the receptor molecule, because here the whole device is representing a biosensor device. Where, yes, madam. Am I? So here the whole device is displayed by here. Main component is the transducer component, and this is electronics where amplifier and the display units are integrated. This transducer is the one we are going to integrate with the electronics, and this electronics where you can integrate your whatever the process you want to generate or monitor, and you can integrate with your internet of things. This will transmit the signal to where you would like to, like doctor's desk or to your mobile. Like that you can transport the signal, and further you can make the analysis. So here the role of transducer is the one. Here the role of transducer is convert the biochemical reaction between the two biochemicals. Reaction occurs in the solution, and it senses the reaction and transduce it, or it transforms the signal to the electronic device, and it transmits to the further for the display and the analysis. If you classify the biosensor, because most of the time for chemical reactions, for chemical sensors, we directly take the chemical into a solution and we put the transducer. So direct interaction between the analyte and the transducer generate a signal. But if you look for developing any biosensor, or macromolecular sensor like DNA or immuno sensors, where it requires antibody or DNA, you have to attach the reagonation layer, and this reagonation layer reacts with the analyte where you have a specific analyte to interact with the reagonation layer. That reagonation will be transduced by the transducer. So based on the receptor and the transducer, you will have different class of biosensor. Based on the receptor, if you use the enzyme, it is called the enzyme biosensor. If you use antibody and antigen interaction, immunobiosensor. If you reagonation layer is like DNA, and we call it as the DNA biosensor. Depend on the, sometimes nanoparticles could also be a receptor, so we call it as a nano-based sensors. On a transducer also you have several types of transducer, mainly 4 classes. One is the electrical transducer, another one is the optical transducer, and thermal, and of course mass based. This gravimetric and acoustic all comes under the mass based transducer. And here we focus only on these 2 areas, where electrochemical and optical based sensors, where electrochemical means electrical or electrochemical, because when we talk about the term electro, means we can measure either change in the current or potential or resistance. These 3 parameters can be monitored using the electrical or electrochemical. Whereas in optical device, you can use, you can monitor change in the light intensity, or the change in the light reflection angle, refraction property. All those parameters can be used used in the optical based biosensor. Again, if you go into subclasses, you have in electrochemical itself there are several techniques, and in opticals again there are several different technologies are available based on your requirement. So here, what we mainly focus is on first electrochemical technique. As I said, in electrochemical technique, there are several types of techniques available. One is the prime technology is the one, this is the one cyclic voltammetry, and this is the electrochemical impedance spectroscopy. These 2 technologies are being used everyone to characterize the sensing behavior of your sensor or transducer, because the electronic part will be bought from the company, and this brand only you are going to develop a transducer which is able to sense the very minimum concentration, like below nanomolar, femtomolar, attomolar, even below that. Or sometimes people are working on detecting the single molecule or single chemical species which is present in the target solution. So here, after developing the transducer, this will be characterized by using the electrochemical as well as sometimes along with the because the electrochemical instrument will give you only change in the potential or resistance or current. But if you want to look at the morphological phenomena or morphology changes, when you are developing a transducer, then you have to use the techniques like electron microscopes, where it's a scanning electron microscope, or transmission electron microscopes, atomic force microscope, or tunneling microscope. All these microscopes are required to look at the morphological changes that your transducer surface is having. Based on this surface property, also your recognition or the sensitivity and the selectivity of the molecular sensing can be tuned. So here in electrochemistry, these two are cyclic voltammetry and the impedance spectroscopy. In cyclic voltammetry, we generally scan the potential from initial to the final, and you get some redox process. Why? Because your molecule or the biomolecule undergoes some electron transfer that will be indicated by your cyclic voltammetric technique. But the same process can be monitored by EIE, electrochemical impedance spectroscopy, where this charging or capacitance current is quite minimum, so you get maximum sensitivity when you are using the electrochemical impedance. Other techniques like amperometry, this is the technique we are going to discuss about along with your glucometer sensing, glucose sensing, where you are applying some potential and you are monitoring the current. And QCM, quartz crystal microbalance, it is the technique where change in the molecular mass or weight on the surface indicated by the change in the frequency of the oscillator, the quartz crystal oscillation. So that will be indicated. And the DPV is another supplementary technique for characterization of because it has more sensitive than these two techniques. So depending on the technique, you can depending on your requirement, you can select the technology. And if you look at the transducer material, various transducer, generally all conducting materials can be act as a transducer material here, which can be used for sensing the analyte present in your sample or blood sample or whatever the target sample is given to you. So when you put the analyte on your transducer, the reaction will occur. So here for the type of transducer, all the conducting semiconducting materials, even the conducting polymers are available for analyzing or getting for using as a transducer. Now people are always interested because we know that technology. Nanotechnology, where people are generating or preparing the transducer material at a nanoscale, and because when you are changing the size or when you are reducing the size of the material, your physical and the chemical properties of the material will be changed. Based on this phenomena, because the lecture which was given by Richard Feynman in 1950s, where you can see that the topic is on plenty at the bottom. That means when you go into deep into up to the smaller scale, you can see large change in the activity of the materials. So based on that, a lot of transducer materials were being developed. And here I just given the 1 0-dimensional, 1-dimensional, 2-dimensional, and 3-dimensional. And these are the dimensional based classification where you have the particles or films or sheet like things. These are the material, like these are all metals, carbons or polymers. All the materials can be prepared in the morphology as you wish. So here, so once you prepared for preparing this one, you can use top down or bottom up method. Those things you might learn in your BSc or MSc classes. So here, once you prepared the material, then I am going to use it as the transducer material. Because you are preparing the nanomaterials, handling the nanomaterial itself is quite difficult for sensor application. Why? Because it is in the nanoscale, and handling by hand, it is also very difficult. So you need to have always a support system to monitor or to measure the behavior of the nanoparticle, or like conducting behavior or thermal behavior or optical behavior. You need to always have a solid support. So here, generally in all laboratory, we take one electrode material, we call it as a glassy carbon electrode, or this part, this is the electrode part, this may be a glassy carbon electrode, or it may be a platinum electrode. So, or gold electrode. These are the three electrode systems being used in all laboratories. For example, if you have this is the solid wire-like electrode embedded in a peak material where it provides the non-conducting insulation. Only this area is going to be reactive part. On top of which you put your nanoparticles, nanomaterials like a carbon nanomaterial or metal oxide or polymer nano or the composites. Whatever the material you prepared, you put it on it. That means you have to attach it on the surface, because nanoparticle cannot be handled separately by hand. So either you take it in solution or you have to put it on the solid surface. So that when I drop it, it forms a thin film, and on top of the thin film, we are going to have your biological or chemical or whatever the target you are going to monitor. This process we call it as the electrode modification. Generally we call it as the, in electrochemistry, electrochemical modification. After modification, I take this electrode, I put it into a solution, and then I measure the the electron transfer. So first thing, this is the basic idea. So in electrochemical sensor, I already told you the glucose sensor is the one which is known to everyone. This glucose sensor was reported, or the principle of glucose sensing was reported by Leland C. Clark in 1962, and he proposed the measurement of oxygen concentration in blood using electrochemical method for diagnosing glucose level in a blood sample. So here, what he proposed in 1962, and this work forms a stepping stone for further development of biosensor and further modification that will lead to occupy the medical market, where just you look at the why, because diabetic glucose sensor is one important because it is related to monitoring the diabetic status of a person. Diabetes in a normal person, for the glucose, or we call it as sugar in general, that is about 80 to 140 milligram per deciliter. If this is the normal range of glucose for a healthy person, if it reaches beyond the 140 milligram per deciliter, hyperglycemia, or if lower than the 140 milligram per deciliter, we call it as the hypoglycemia. So here, why? Because if it goes beyond, this leads to a lot of organ failures like kidney failure, or increases in blood pressure, or you get some other kind of diseases related to skin, or you will have some other neurological disease also being formed along with your diabetes. So now people are afraid about the presence of excess amount of glucose in a blood sample. So people are trying to monitor or trying to control the amount of glucose present in the blood. And why diabetic forms? How it forms means because your glucose, when you take food, it is go through your intestine where it is digested, and the sugar is transported to a blood cell by a transporter called insulin, and the insulin takes the glucose which is flow in your bloodstream and take it to the cell surface, where it go into the cell membrane, and it involves maybe ATP, ADP conversion, and it gives the energy. So if this process, if the transport process is stopped means, or if it is slower means, your glucose is accumulated in your blood, and that leads to the inception of the diabetes. That again leads to several symptoms like the frequent urination, being high, always feeling thirstiness, and feeling of hungry and tiredness, whatnot. All kinds of diseases are related to the presence of excess quantity of glucose in your sugar, in your blood. So this can be measured. The principle which was proposed by Leland Clark was by measuring the change in the oxygen concentration, because this glucose is normal sucrose is converted into beta and LD glucose, and only beta D glucose is specific to glucose oxidase, and this in presence of oxygen it reacts to give gluconic acid and hydrogen peroxide as the byproduct. Where your glucometer works based on the principle of analyzing or quantifying the hydrogen peroxide concentration, which is generated from glucose and glucose oxidation reaction. So here, this molecule, gluconic acid, which will not undergo oxidation or redox process under the potential or below the potential 700, and but the hydrogen peroxide is the one which is electroactive at this potential, which is monitored at a platinum electrode, on top of which your glucose oxidase can be interacting. That means the reaction between the glucose and glucose oxidase is sensed by the platinum electrode through the analyzing the hydrogen peroxide concentration at an applied potential 600 to 700 millivolts. So why here, when you look at the market product glucometer, it looks like, sorry, I don't have the picture. Let me check. Okay. I have shown the picture previously. This is the one glucometer. Sorry, here it is a chip-like thing where your meter is here. Once you insert the chip and it switches on, as soon as it switches on, it applies the 600, 700 millivolts. It is fixed. That is a constant potential is being applied. And once it is applied, and then you are putting a drop of blood on it, and you get a reading on it. This is a kind of, it is a electrochemical measurement, and the technique which on which is this system works is chronoamperometry. That means where we are applying potential and we monitor the current. This current was generated because of the reaction between the glucose and the glucose and glucose oxidase, which was attached to the electrode surface. For attaching the glucose oxidase, here is the basic thing where you take membranes, and in the polymer membranes, one is cellulose acetate and another one is the polycarbonate membrane. Between that we put glucose oxidase. These two membranes are semi-permeable, where the top membrane polycarbonate, which is permeable only to glucose, and here the hydrogen peroxide. Yes, madam. Madam, am I audible? So here the cellulose acetate is permeable only to glucose. So whatever the reaction is, only hydrogen peroxide is sensed at the platinum electrode. But this sensitivity depends on how you are immobilizing the enzyme on top of the electrode material. For that or sensor material, where you have different types of immobilizing chemistry. One is covalent attachment, where you have the material or this conducting material, you create some functional group and you make covalent attachment. Or you use some linking agent, cross-linking agent like glutathione or EDCNHS, like coupling molecules to couple the enzyme on the surface. And the entrapment, we are using the polymer for the entrapment. Adsorption, this is the physical adsorption. Our affinity is also by electrostatic, molecular affinity, because once we have hydrophobic or hydrophilic, based on that property also molecules can be absorbed on the surface. These are the methods proposed for attaching the enzyme on the glucose oxidase enzyme on the sensor surface. So here is one example where the Itamar Welder from Israel, what he proposed was right. So here this because what I told was we put this enzyme on the electrode surface because your active site NADH2 NADH2, this is the active site which is buried inside the cell or on the center of the enzyme molecule, and where it undergoes electron exchange between the glucose and the enzyme. But this electron transport is quite difficult to sense. For that, people are using nano particle or nano materials, some polymers or metals or metal oxides or some inorganic complexes to make ease of the transport of electron between the glucose and the active site of enzyme. Based on the material that you are using, these glucose sensors are further classified as first generation sensor and second generation sensor and third generation sensor. So in first generation sensor, we are having the enzyme and we are monitoring the hydrogen peroxide or oxygen concentration. But in second generation sensor, we put a metal mediator like complexes like ferrocene complexes, ruthenium bipyridyl, or some inorganic and organic metal complexes to mediate the charge transport between the glucose oxidase and glucose to enhance the sensitivity of the detection. Why we need to have the sensitivity? Because for diabetic, it is 80 to 140 milligram per deciliter. If you convert that into the millimolar, it is 0.3 to 3.3 millimolar. And so you have to develop a sensor that works or that indicates a change in the concentration of the analyte in the range where you need to monitor. So here, this group, what they propose is they use the gold nanoparticle along with the glucose oxidase to promote faster electron transport between the gold nanoparticle and the enzyme, and that will be again transported to the solid conducting material on top of which these two are attached. By using this, they are able to sense different range of glucose, and the saturation limit is obtained about 100 millimolar, because your diabetic scale is already falls in this range. This sensor can also be used for diabetic sensing. But why? What is the difference between the markedly available sensor and here? In market sensor, they use some metal complexes. Rather, here they are using the gold nanoparticle as a mediator. This is how they are trying to improve the sensitivity of the sensor. And also, when you are having the nanomaterial, this also provides biocompatibility for the glucose oxidase so that its stability can be increased. Because the storing of the enzyme for longer period of time is also questionable. So when you want to enter into the market, you have to provide a good biocompatible nature so that the enzyme should not undergo any degradation. For that, people are trying to work, trying to develop or trying to integrate more biocompatible nanomaterials along with the enzyme so that the enzymes chips can be stored for years or months together till it is sold. So here, so what I am trying to say is try to use some nanomaterial so that you can enhance the detection rate. One is the nanomaterial or nanometal, gold was used, and here they have developed manganese-based pyrene pyrene complex, which is undergo reaction in presence of glucose oxidase. And if you look at the linear range or the exchange of linear range between the linear range obtained is 50 to 550 micromolar. You can ask here, because the concentration is very low, this cannot be used for a diabetic sensor, but it is not like that. But the question is, even if a smaller change in your glucose level, like if I have 140 milligram per deciliter, if some other person is having 140.5 milligram, then that 0.5 milligram should be quantified exactly. For that purpose, people are trying to increase the sensitivity of the sensitivity can be calculated. How you can calculate the sensitivity is nothing but the slope of this linear portion of the curve. This we call it as the calibration curve, and the slope of this curve gives you the sensitivity. When you have a higher sensitivity, then smaller change in glucose concentration can be indicated by your device. That is why people are trying to use nanoparticle or nano complexes like inorganic or organic complexes. In another recent advancement is by using the carbon nanotubes and graphene or carbon dots or metal quantum dots for integrating along with the glucose oxidase to monitor the glucose reaction. Here, one of the material, carbon nanotube, on top of which glucose oxidase is attached by covalent bonding. This attachment, either covalent bond or you can attach it directly by hydrophobic interaction, because this molecule is also hydrophobic and this surface is also hydrophobic. It can absorb through hydrophobic interaction, but the stability is quite questionable. So people are trying to attach the glucose oxidase by covalent attachment, and they try to monitor. And after attachment, it will be again put it on the solid transducer for further investigation. And here, the linear range, what they have reported is about 1 micromolar to 5 millimolar. And the lowest detection limit, which is lowest detection limit, is the one, the lowest concentration that your sensor response, that is about 0.36 micromolar. And so, of course, recently we have also attempted using carbon dots and the gold nanoparticle and the zinc oxide nanoparticle along with the enzymes. But we are not much interested in using enzymes. Rather, we try to develop a glucose sensor which avoids the glucose oxidase. Because why? Because attaching the glucose oxidase on the sensor surface requires RS chemicals, and also it requires tedious protocols to attach, and the disposable of materials which are resulting as a waste from the reaction. Considering all those things, and also one more problem is the stability of glucose oxidase is questionable when you would like to store the sensor device for years together, because now for going for market, your sensor should be at least stable for 3 months. Below 3 months, the people will not like to buy your device because it cannot be sold immediately. So for that purpose also, we try to eliminate this glucose oxidase by developing non-enzymatic sensors, where we directly monitor glucose oxidation at a metal, because carbon materials will not undergo oxidation because of a requirement of higher potential than your metal surface. So we try to use the platinum, gold, especially these two material, along with other transition metal like cobalt, nickel. All materials which are conducting can be used for monitoring the glucose reaction directly without using any enzyme. So here, how the reaction occurs, the sensor metal surface generates oxide hydroxide group, which interacts with the glucose, and it undergoes the glucolactone or gluconic acid what are the product which is formed during the glucolactone or gluconic acid or hydrogen peroxide. And here, for example, if you look at the application of platinum, this is the solid line is the reaction of glucose on platinum, but this peak is not highly reproducible. One important parameter of sensor for medical application is that your sensor should be accurate. And whenever you use a sensor, it should show the same value. Then only it can be applied for the diagnostic, or accurately you can diagnose the disease. But here, once it complete the cycle, if you come back to the second cycle, it almost becomes zero, because the carbon monoxide, because these platinum are material which oxidizes glucose into carbon monoxide and water. This carbon monoxide will strongly absorb on the metal surface. So direct sensing of glucose at the metal surface is quite difficult. So what we did was we took we prepared ruthenium oxide by thermal annealing from ruthenium chloride, and it is mixed with the tetrahydrofuran solvent, and then it is put it on the electrode surface, and it forms a ruthenium. Of course, this mixture was consist of ruthenium oxide and polyvinyl chloride as a binding material, because without binding, the stability of the ruthenium oxide on the surface is quite difficult. So we attached this, we integrated ruthenium oxide along with PVC, which is completely soluble in tetrahydrofuran. By mixing this, we form and drop tested, it form a film, and this film is reacting with the glucose to produce glucolactone, or it undergoes further oxidation to give you the carbon dioxide and water. And here we monitored glucose sensing in alkaline medium. Remember, your glucometer works with blood sample, which is having physiological buffer 7.4. But here, this material, even in the previous slide, what I showed was the platinum. This platinum or metal, in general metal or metal oxide, which are very active in higher alkaline pH, greater than 10 to 11, so 10 to 12. So this is the pH range where you can sense glucose or oxidized glucose effectively at the metal or metal oxide. So we try to investigate the glucose sensing using the sodium hydroxide, and also we want to extend it to the physiological buffer, so that we prepared a phosphate buffer saline solution, and we try to monitor the glucose sensing. Here you can see this black dotted line is the sensing current in the sodium hydroxide solution, whereas this red dotted line is in the phosphate buffer solution. Remember, the activity is quite lower when you decrease the pH of the measuring solution by 7 or 7.4. But we want to apply it for blood sample analysis. So what we try to do is we always characterize the system in phosphate buffer, and we see this, we taken this potential as this is the maximum potential, and this potential as the constant potential for chronoamperometry, and we monitor by increasing the concentration of glucose by drop-wise manner. And you can see this linear range, we get around 100 micromolar to 107 millimolar. But here the problem was these results cannot be reproduced all the time. Now, you can reproduce, but you cannot reproduce exactly. That is the problem, or accurately. That is the problem with this electrode. But based on our principle, we got this project from ICMR to develop the non-enzymatic glucose sensor. That was the project we got. We developed the device, but still we are not able to get 100% reproducibility. So still work is, we are working with it with different materials. And as we are also interested in using carbon material, here we use the carbon nitride, and on top of which we anchored platinum, because carbon nitride itself cannot sense any glucose sensing, and therefore we integrate or we modify the carbon surface using platinum and as well as the zinc oxide. Why we need to attach platinum and zinc oxide? To enhance the reusable behavior of the sensor. By this time, by applying the carbon material and along with metal oxide, we are able to reproduce in a whole blood. We can get the signal only for 4 times, whereas if you use the serum, we can get more number of times it is reproduced, but still it is not enough for the market product. So still we are enhancing, because in the original glucose sensor, they are used polymer membrane to entrap glucose oxidase. But here we did not have any polymer membrane. Instead, we have only the carbon nitride, platinum, carbon nitride modified with zinc oxide and platinum. And we measure the, without glucose oxidase, we are able to sense the glucose in a blood sample. And this one was again characterized by various techniques. As I told you, you have to look at the morphology. Of course, if you want to good publication, there are 3 M's are important, like SEM or TEM, other spectra AFM, like people are calling it as the 3M techniques. Why, if you don't show these kind of morphological features, or you develop a new material, or if you are not able to claim that you have prepared a new material, then your paper will be getting rejected. So when you are, as you are a scholar, you should have published a paper. For that, we can use a different material for developing the same type of glucose sensor with using different materials. But only our goal is to develop sensor which is able to give you highly reproducible and highly reproducible, not only in results and also in fabrication and the fabrication process, further process for sensor mass production. So this sensor give a linear range from 0.25 to 110 millimolar. You could ask that here the PVC based sensor showed 100 micromolar, whereas when I use the carbon nitride and platinum, you see this one is 0.25 millimolar. The linear range starts from 0.25 millimolar. The lowest concentration could not be detected below 0.25 because this carbon material also provides some capacitance current where your sensor signal is embedded. So that sensor capacitance current should be eliminated using other materials. So for that, graphene and graphene oxides also we were used. Once you fabricate, you have to quantify, you have to describe all your sensor features, like the linear range. From the linear range, you can estimate the lower concentration and higher concentration limits and the reproducibility factor. And of course, you have to check about the biocompatibility and other interferences. Interference means here along with glucose like ascorbic acid, uric acid, all other amino acids which are all present in your blood sample. So you have to eliminate the reactions of other analytes in presence of glucose. So only you have to selectively detect the glucose using your sensor device. In another application, we used multi-walled carbon nanotube along with zinc oxide. Just RuO2 was replaced here. So PVC is also there. Multi-walled carbon nanotube is also there. This enhances the biocompatibility nature of the sensor material. For example, here we measured the zone of incubation, that means the antibacterial activity, for example, to bacteria, because once you would like to develop the device which is should be integrated, because your IoT devices all based on integrated sensors, which is able to transmit the signal from your body to your mobile or your electronic device, where it can be processed for further application or further diagnosis. So for that purpose, whatever the sensor material you are going to develop, you have to evaluate biocompatible nature of the material so that you can integrate your sensor device on your body, because these are all wearable sensors. So where you have to where you have to study all the biocompatibility nature of your materials, so that it will not induce any disease when you are wearing those devices onto your body. So here this is the antibacterial activity, and this one is the hemolysis, blood cell degradation. All these shows that integration of material along with PVC enhances the biocompatibility as well as the antibacterial activity. And the linear range again is down to 20 micromolar to 17.8 millimolar. Here my point is, you just change the material and the combination of material, or we call it as the composite. Like metal can be composited with the carbon material, or metal can be composited with your polymer material, or other semiconducting material. All those materials can be used for developing any type of sensor, not only for glucose. You can also develop sensor for other metabolites like neurotransmitter metabolites, or other metabolites like uric acid, creatinine, all other chemicals which are responsible for type of disease you are going to measure. You can integrate, develop the sensor. So here, if you talk about Internet of IoT integrated sensor devices, as far as glucose is considered, there are 6 types of glucose monitoring devices available. One is the continuous glucose monitoring system. This is a device which which measures the glucose concentration at a subcutaneous level. That means it is like a watch, glucose watch. It can be wearable on your hand. Once glucose reaches to your sensor part, it indicates the signal. So this one is the one, and another one is this collects the glucose data 4 times per day, or you can tune your, or you can fabricate a device so that you can monitor at every half an hour, or every 1 hour, or every the time duration, whatever you like to, and it is integrated onto your body, and it stores the signal, and it stores the signal into the hospital system, where doctors can use the signals to analyze a particular person's glucose level. And another one is Bluetooth low energy implantable glucose monitoring system, where this one is the data collected by this device is transmitted through Bluetooth to your smartphone or iPad, where it can undergo signal processing for visualization and for making the decision. And another one is implanted sensor devices and modules, like here, where you can have your glucose sensor integrated into your body. Whenever there is a change in the glucose concentration, it can monitor your disease by, if your device is also integrated with the insulin system along with your glucose monitor. When there is a change in the glucose higher concentration, it releases the insulin into your body so that your glucose level is maintained. Another one is non-invasive. These are all non-invasive. Actually, non-invasive. The other one is it is electrochemical system, where you wear your device on your body. When glucose reaches to your system, it again monitors the glucose and glucose oxidase by amperometrical technique. But you can also use optical devices for monitoring glucose system, where near infrared light can be used to monitor the glucose level when it is attached, when it is changing the glucose occurs on in your body. Another system is glucose level monitoring system based on wireless body area networks for detecting. Here, this system built using glucometer sensor and other modules which are required for analyzing the signal. Doctors and caregivers can access the web page and they can monitor the glucose concentration on the patient remotely. And so these kind of systems are being developed for glucose sensor, where Internet of Things is already associated and it is in the market. Here, this is the picture which is tells you this is the glucose sensor. This kind of shirt where your glucose sensor is integrated, and all the vital signals. The vital signals means here the vital signal. Like body temperature, pulse rate, heartbeat rate, and respiration rate, blood pressure, along with glucose concentrations can all be monitored using by integrating your sensors onto your body using some method. Here is the shirt developed by nanomaterial, which is also integrated with glucose oxidase and other materials. Once you put it on your body, it measures all your vital signals. When there is a change in these parameters, it transmits the signal to the doctor, where he can advise to take medication. This is how your Internet of Things is developing along with your glucometer, along with diabetic monitoring especially. People are also interested to develop not only for glucose sensor, but for other diseases, where we mainly focus on neurochemical disease, neurochemicals like serotonin, histamine, dopamine. These are all the various molecules, neuromolecules, which are actively involved in transport of neurons from one cell to the another cell. In other words, cell to cell communication, all your cell process will be controlled by these neurochemical chemicals, which are exchanging between two cells. For exon and other things, this terminology you can look at the internet. This synaptic vehicle, which are carrying the neurochemicals, which is released at the junction between the receptor cell and the donor cell, and the exchange of which, if it happens to be higher concentration or lower concentration, that leads to neuro diseases. Type of neuro diseases is like feeling of sadness. Of course, when you approach a doctor having some kind of these are all the symptoms where your doctor asks you, and they correlate along with all these other parameters like lifestyle parameters, where how you feel, or illness, worthiness, emptiness, all the irritation or restlessness, all these they correlate, and they try to monitor the chemical concentrations which are present in blood or urine. Then they correlate what is happening into your body. So for that also people are trying to develop a sensor in our laboratory, because I'm not going to talk you about all the sensors which are available, reported in the literature or available in the market, but I will focus only on my laboratory result, where we try to develop, for example, here a dopamine sensor, along with acetaminophen, melatonin is the one, acetaminophen is the paracetamol tablet, paracetamol and melatonin, these two interact, these two interact which give you product that will affect the concentration of dopamine. This is how we are trying to understand the neurological chemical behavior or the change in the concentration of neurochemicals in blood and urine, so that the disease can be diagnosed exactly or accurately. Here we prepared iron oxide by hydrothermal method, and it is decorated. It is doped with platinum. This is doping was done by physical absorption, where the most of the platinum are attached on the surface. Sometimes, because it is the whole material is subjected to thermal annealing, sometimes it may diffuse into the interstitial site, or we call it as the interstitial doping, and we try to monitor the signal. Here, this is the sensor material, and this is the this is the transducer without this material on top of it. Once we add this material for monitoring dopamine, acetylcholine, or melatonin, you can see that increase in signal. Here, our aim is to detect individual molecule as well as simultaneously all the three molecules together in a given solution. So here, dopamine undergoes at a lower potential compared to the acetaminophen, which is lower than the melatonin. So all these chemicals, when you are changing the potential, these will undergo the oxidation and the reduction at different potentials. So you can quantify, because the increase in current is directly proportional to the concentration of the analyte present in the given solution. So here, and of course, once you develop the material, you are going to characterize using the all other techniques, supporting physical techniques like X-ray diffractometer or scanning electron microscope. Here we try to understand the formation of composite between the iron oxide and the platinum. So here, so platinum formation will be identified by the appearance of XRD peaks, which is reference, because all these peaks are referenced to standard reference data available in the data bank. With reference to that, we can come to a conclusion that the composite was formed by using the iron oxide and the platinum. So here, this is the morphological features, and once you get the linear range and other things, you can correlate that some morphological features can influence the deduction limit or the linear range like that. So here, this is the simultaneous sensing of all the three analytes together are different, because once you develop a new material, then you have to distinguish between each component's contribution. That is why you have to make all the measurements for each component which constitute a composite. And then you have to estimate the linear range and the lower deduction limit. In our study, it shows the 0.1 nanomolar is the lowest concentration of the dopamine. And here, this is the linear range up to which your current is increasing in concentration. So in another application, again, as I told you, when you change the material, you are going to change the linearity or the sensitivity. Sensitivity, again, this is the slope. If you yes, sir, yes. Here, yes. Am I audible, sir?
Yes, sir, you are audible.
Sir.
Okay, sir. How long time, sir, I can take? Another 1 hour?
Yes, sir. Yes, sir.
So here, we changed it to using the perovskite material and modification with graphene to form a composite. And again, we measured the dopamine. Here you can see that the nanomolar concentration range, linear range, 72 nanomolar, whereas in the previous one, it is around 1 nanomolar. So if you look at it, when you change the material, perovskite, transition metal oxide to the perovskite, gadolinium, the rare earth material coupled with the transition metal to form perovskite and modified on the graphene surface. So here we measured again dopamine, acetaminophenol, acetaminophenol, where the morphological changes results into the LOD is around, this is micromolar, 0.05 8 micromolar to acetaminophen and 0.096 to dopamine. This is how we try to develop a sensor, but these were evaluated in urine and blood sample. We collected urine and blood sample and we try to measure the concentration of dopamine, especially the dopamine in the real sample. But in most of the time, getting the real sample is quite difficult. For that purpose, we use a standard addition method, in which you take the urine sample, whether you don't worry about whether you have, but most of the sensor development laboratory are using this method. That is why we also followed this one. We try to add the standard material, standard chemical which is applied from the vendor into the urine, and we measure the concentration which is present. But this method is not exactly applicable for the physiological measurement, where you have to get a sample from the diseased or neuro affected person to quantify the signal. Because we could not able to get those kind of samples, we try to use the standard addition method. For the sensor characteristic, this is enough for getting publication. And another method, we also used the binary metal oxide, manganese oxide and titanium oxide. Again, it is modified on a graphene, again used for dopamine sensor. Here we try to understand how these metal oxides interact with the graphene oxide. Here 0.5 millimolar dopamine, you can see this, the composite of titanium oxide, manganese oxide on graphene showed a higher activity for dopamine sensor. And the linear range, again it falls 0.01 nanomolar, and the linearity range is around it is around 100 nanomolar only. It is 0.01 to 100 nanomolar. After that, it reaches the saturation limit. So this is how these materials were used for developing sensors. Especially we are interested for developing non-enzymatic sensors. So we try to avoid all enzymatic reaction, and we are developing glucose sensor as well as neurotransmitter. Along with these type of sensors, we are also trying to develop diagnose the plant disease based molecules like acetylcholine or other like chlorophyll content. All those material molecules can also be sensed using the conducting transducers, developing using the nanotechnology. But if you don't use any nanomaterials, the sensing of these lower quantity of the material, quantity of the chemicals is very difficult, and most of the time you don't get good signal without using nanomaterials. In another area, my laboratory focusing is on developing DNA-based sensors for diseases diagnosis. And here our laboratory is mainly focusing on pathogens as well as cancer-related DNA molecule. Why? Because all the diseases, all the non-communicable diseases like DNA-based diseases occurs because of change in the DNA mutation, the change in the DNA base pair. So here, for example, I forgot to have the picture here, DNA strands. You know that we already know that DNA consists of 4 bases like adenine, guanine, thymine, and cytosine. and these these bases are interlinked using phosphate backbone and sugar sugar linker between two bases. When one single stranded DNA interacts with the other DNA, it forms a Watson and Crick base pair double helix. This double helix is the one is responsible for generating or for producing the protein molecules, proteins, which is essential for the body functioning. If there is any change in the mutation, for example, if the DNA formation or double helical formation is not following or not obeying the Watson and Crick double helix rule, that means adenine can bind only with the thymine and guanine can bind only with the cytosine. Of course, there are hydrogen bonding between these base pairs. If there is any misplacement, a misplacement of DNA bases, for example, if I wish to have adenine to thymine, if I replace thymine by cytosine, this adenine cannot react with the cytosine. So that the process, the transport process or the heredity information transport is prevented, and it results in the synthesis of wrong protein and leads to the different diseases like cancer or other non-communicable diseases. And recently also we have some COVID related sensors which are all which is now on market. It is based on the Immuno sensors and the DNA based sensors. We'll talk about later. So if it does not follow the double helical rule, then it leads to the diseases. So where we try to analyze the DNA strands, DNA concentration of DNA in the given blood sample or given tissues. Why? Because once you go to the doctor, they will collect your tissue or cells, and then they extract the DNA, and then they try to put it on the, try to analyze whether there is a formation of double helical strand in your sample, so that or if there is a perfect double helical formation or imperfect double helical formation, to come to the conclusion. So for that, two different methods mainly applicable. One is a optical method and another one is the electrochemical method. In both the method, we try to analyze the formation of double helical strand on sensor surface. If there is a complete perfect double helical formation on your sensor surface, you will get a good signal. If you don't have double helical formation on your sensor surface, you will not get any signal on the signal from your sensor. If there is a mutation, single mutation is present, then how about the quantification of the mutation? Hello? Sorry, sir.
Sorry, sir.
Am I audible or no?
Yeah, sir, you are audible, sir.
Okay, okay. So here in optical system, we use fluorescent based sensors where excitation, we use dyes, two different dyes. One is Psi3, which gives a green light, and another one is Psi5, which gives the red light. So doctors, what they do is, because all the diseases related to DNA mismatch is already analyzed, and it was data bank consists of data bank, we call it as a NCBI data bank, where you get all the DNA sequences related to all types of cancer or all types of neurological diseases like Parkinson's disease or Alzheimer's disease, whatever the disease you would like to. If you look at the DNA sequences responsible for those diseases, which are all analyzed and kept in the NCBI data bank. So these DNAs are all diseased DNA sequences. Remember, the data bank give you diseased DNA sequences. Now I get a person I get a person who is, for example, cancer patient. I get his blood sample or his cell or a tissue sample. I extract the DNA and I put the DNA on the sequence which I taken from NCBI data bank and try to see whether there is a formation of double helical DNA on the surface. This is the method of forming or using the sensors for diagnosis. That means you have the sequence already. You in your hand you have a DNA sequences for a respective disease, and it is attached to your sensor surface. Now I get a person which is who is also having some disease whose sequence is matching with the sequence which I am having already. So if these two sequences are matching, then you can do it. For analyzing this signal, it is very difficult to say that whether the person. For that we use labels. Because when I, for example, if I go, this is the process. So here you have a DNA. These are the labels which are going to use it. I take it one for the this sequence obtained from the, for example, data bank, and another one label which is also going to use it for the DNA sequence obtained from the patient. So I use two different labels. One is Psi3 and Psi5, which is excited at two different wavelengths, and you get emission spectra, which is red in color and green in color. Here, for example, this is the protocol where people are using to extract the DNA. That cDNA is a complementary DNA. That a complementary DNA, or sometimes they will also call it as a chromosomal DNA, because DNA is expected. So here, there's some loss is coming. So here, after extracting this DNA, I label with the label, dye label. So one DNA you get Psi3, and one DNA you get a Psi5. So if you assume that, so these two DNAs are put on the sensor surface. On this sensor surface, I already have a sequence which corresponds to, so for example, red in color. If I put these two, put the DNA from the extracted from the patient, if I get the whole red in color signal, then the person is diagnosed as having the DNA sequence which is immobilized or attached to the sensor surface. That means these two DNA are corresponding to the same disease. That means the person is having the same disease. Like for example, if I put a cancer, for a colon cancer means, if I take, for example, if I take colon cancer sequence and I obtain a colon sequence from the patient, label with, and then I get a red signal, means you get your disease control. Sir, I think too noisy signal. Someone is keeping the mobile phone near to your device, or here. So in sensor fabrication, we take a solid polymer sensor. We take a solid surface and we attach a single strand, and strands or target strand which are labeled with dyes, and it is reacted with the single strand to form a double strand according to helical rule. And once it is based on the color, based on the color, I am going to diagnose or label the person as a diseased person. This is the one of the technique proposed by DNA microarray technology, where they get, for example, here. For example, here I get a diseased sample on the surface. I get a normal person and a disease affected person, tumor affected person, for cancer person, and I get DNA from both. Now I extract the DNA and label with my labels, and then put it on the sensor surface and try to use the fluorescence spectroscopy to analyze the image, fluorescence image. Here, the red spot indicates the signal from the tumor or the cancer person, and the normal one, that is the green spot, is from the normal person. So the red spot indicates the person is affected or getting disease, whereas the normal person does not. Green signal indicates it is the normal sequence and the patient does not have or affected by a disease. This is how fluorescent based diseases are being developed. And here in electrochemical method, this one is the optical device where you require two lasers to excite two different dyes like Psi3 and Psi5, and you get to compare the intensity of the green and red signal, and you use some bioinformatic tool to come to the conclusion that whether the intensity of the disease. And here we try to use the electrochemical method where we try to put the DNA on the surface. For example, this is the DNA. I take a gold electrode, I put a gold, and the DNA can be attached to the surface either covalent or electrostatic interaction or by physical absorption. But we usually prefer to have a covalent bonding because we need to have a strong bonding in order to avoid the stability problem. So here we take a gold surface and we put the DNA which is having thiol group. This thiol and gold is very quickly react in a nanosecond scale, and it forms a covalent bond. That means it can attach, we attach with the DNA on the gold surface. So here again, similar to your optical device, whether in electrochemical, you can measure the attachment of DNA on the gold surface by using cyclic voltammetry or impedance or whatever the technology you wish to. So what you get is, for example, here attached to the single stranded DNA, when I put the target DNA, it forms a double helical DNA, and the current before and after the target interaction, you can see that there is a change in the signal intensity, or the potential of the peak is shifted towards the positive or negative, depending on your reaction. So this is the basic principle of using electrochemical sensors for DNA molecular analysis. What if I have a mismatched DNA in my hand, or what if there is no bonding between either one base or two base, whether we can correlate the single base or single nucleotide single nucleotide morphism. Yes, polymorphism like SNP or a double mutation, triple mutation like that. This is that is why. But for amplifying the signal, two different strategies are using. One is because if I put a double stranded here, the change in the intensity is very, very minimum. For that, we try to attach in the target DNA, target molecule, we use some label like enzymatic enzymes like alkaline phosphatase or radis peroxidase. So here what we do is once I get to the extract of the DNA, I attach this enzyme on the surface, and then I take it to react on the single stranded DNA, which is previously immobilized on the target previously immobilized on the gold electrode. Once I do that, what I do is, because this is an enzyme, if there is a perfect helical formation, this enzyme is present on the surface. If there is no helical formation, means this enzyme will be absent. So if enzyme is present, then I put its substrate, it undergoes a reaction at a particular potential, then it creates a signal with this product, which is the para-aminophenol phosphate, which releases para-aminophenol, which is electroactive, whereas this para-aminophenol phosphate is not electroactive. Once this substrate is released from the enzyme reaction, it undergoes electrochemical redox reaction at a given potential. For example, in this system, we have applied 300 millivolt so that it can generate the signal. So here, once there is a formation of the double stranded helical formation, you get a signal increase. If there is no helical formation, you get only no increase in the signal. This is how the DNA molecule can be sensed by using enzyme labels in electrochemical technology. And here again, use of enzyme again is quite a problem, because you have to think about attachment chemistry and then stability, and this reaction requires a particular potential for its substrate activity at the gold or the sensor surface. So in order to avoid this, our laboratory is mainly focusing on non-enzymatic molecular DNA molecular sensing, where we directly put a single stranded DNA and put the double stranded target DNA, and we form the double stranded DNA, and before and after you can monitor the signal magnitude changes between the single strand and the helical formed double stranded DNA. For this purpose, you need to have 4 steps: immobilization of the transducer and hybridization in the target, and the conversion of this hybridization signal into electrochemical signal. And for this, sometimes what we do is, even for this one, if you apply different range of potential, the bases which are present in a DNA, like guanine and adenine, will undergo oxidation. For example, guanine undergo oxidation at 0.8 volt, whereas adenine will undergo at 1.1 volt. And recently, by using the graphene or other carbon material, where you can see all the 4 bases will give different oxidation peaks. That means when you put a single stranded, this is the signal. When you have a double stranded, you have an increase in number of amount of current signal that is directly proportional to the amount of base that are present in your strand. But this one we call it as the direct method, where the bases undergo oxidation. But for diagnosis, if you oxidize, this will not give much accurate diagnosis. For that, improving the diagnosis method, and people are trying to use the charge transport through the DNA molecules, because though this is the biopolymer, we call it as the biopolymer, which is able to, most of the time it was claimed as the non-conductive, but recently it is proved that it is able to transport the current through the double helical formation. The criteria required for this transport is that the base difference between these two, the 0.34 nanometer. That means the distance between these two bases should be around 0.34 nanometers, so that the guanine, the pyridine and pyrimidine bases between the 2 bases mingle together, and they are able to make an electronic cloud, and through which your electron charge transfer could occur. If there is no charge transfer could occur, then this metal signal will not be diagnosed. One more minute, please. Sir, now seminar la irukra appo poduva. Okay, thank you. So once, so if the in along with the double helical, I also took a metal complex which undergo direct reaction at the sensor surface. Because this guanine undergo oxidation and reduction, this oxidation current is received by this metal complex, which generates the signal on the sensor surface. This means if it is not able to undergo oxidation, then this reaction will be prevented. That means if you have a single strand in which DNA guanine is misplaced, then this reaction will be prevented. So based on that, recently people, because here this is at the metal electrode, and the recent invention of graphene and carbon nanotubes and carbon materials, they have used to direct measurement of strand sequences where they put the DNA on the surface of graphene by hydrophobic interaction, and they label the probe with the kind of fluorescent label. Because it is undergoing hydrophobic interaction, this fluorescent label is very closer to the sensor surface. Of course, this one is from the optical measurement. It is not related to the electrochemical measurement. Later I skip to the electrochemical part. Here, this capture we put a single stranded DNA labeled with fluorescent probe. Once it is having very closer contact with the graphene surface, the fluorescence is 0.6. After adding the target DNA, this single strand DNA reacts with this target and releases the probe away from the graphene surface. Thereby, it releases the fluorescent signal. That means appearance of fluorescent signal indicates the formation of double-stranded DNA on graphene surface. This is called on signal. In other words, if you do the reverse, for example, if you have a single-strand DNA, if you label the and once if you have only single strand, you don't get without label no signal. If I have a labeled target, once it reacts, it will give a fluorescence, and you have to wash it. If you wash it, the unreacted DNA will go away. If there is no reaction, means the fluorescent signal will not reoccur. So based on this, they have developed on and off sensor device which indicates the formation of single strand and the or presence of single strand or the double helical structure. So if you look at it, again, what we did was we eliminate this fluorescent signal, and we want to use the electrochemical measurement. For that, we attach the graphene surface on top of the gold layer using some molecular intermediate molecular. Here we use the mercaptopropionic acid, on top of which we also put because we want to have a covalent linking between the single strand DNA through a gold nanoparticle. And because adding the gold nanoparticle on top of the graphene or graphene oxide will enhances the reactivity, active reactive sites, so that you can enhance the hybridization event or signal intensity. For that purpose, the combination of graphene and gold nanoparticle were taken together on a gold electrode, because we don't want to have a physical absorption of graphene on gold surface. Rather, we also want to stability of the layer, so that we put the intermediate molecular layer between these two, and on top of which we put a DNA capture probe, and then target probe. We form a helical formation. How the signal looks like is this one, where you have a gold electrode and monolayer based electrode B, and then if we put the graphene layer, then we get a signal like this, because we try to characterize the electronic behavior of the graphene gold nanoparticle composite, we use cyclic voltammetry and impedance in presence of ferrocyanide as the redox probe in the solution, whose reaction is on the modified electrode is monitored. Here it indicates the attachment of graphene and gold nanocomposite on the electrode surface increases the signal because of graphene is attached to the gold surface. And once we attach it, then what we did was we put the strands and helical formation. I lost that PowerPoint in preparation. Where what we get was the signal like what earlier I showed. No, this is the signal difference. The same kind of signal difference we get when you use the graphene and gold composite between the single strand and the double strand. Using this device, we are able to detect the concentration range. I think we got around 10 picomolar concentration of the target DNA. And when we have the base mismatches, the signal intensity between the single strand and the double strand is also varying. I forgot to add that signal here for PPT here. Sorry for that. I try to give in another slide. Similar to that, we also use the lipid molecule. Instead of graphene and gold nanoparticle, we replace this layer using cell membrane, lipid molecules, which consist of, you know, that hydrophilic and hydrophobic head and tail group, and it is a constituent of a cell membrane where it protects the cell content and it safeguards all the cell activities. So here, because this cell membrane can be act as a receptor for DNA, and we try to use, because we are not going to talk about the receptor activity between the lipid membrane and the DNA, rather we are going to attach the DNA on top of the lipid layer, and we try to use it for the diagnostic application. So here, what we did was we put this is the lipid layer integrated with gold nanoparticle, and which was attached on the gold surface using the another intermediate layer, and it is characterized by cyclic voltammetry, where your bar is black signal and red one is your DOTAP. This is lipid molecule, and lipid molecule gold composite. This one is green, and your this one is another. This is directly on the gold. And why we put this molecular layer, intermediate molecular layer, in presence of lipid is also another reason that if we put directly the lipid layer, it will. We want to have a vesicular-like structure. But this vesicular-like structure will be broken if you put it directly on the solid surface. That is another idea of using this intermediate layer between the lipid layer and the gold electrode. After this, what we try to do is we put the single-stranded DNA and double-stranded. We form the double-stranded DNA here. This is the single-stranded DNA. It gives the signal here. When we put the double, when we form the helical formation, we get again change in the or reduced signal. What I'm trying to say here is we prepared different molecular layer consisting of different lipids, like DOTAP is the one. Another one is like dope is another lipid molecule. I don't have the formulas here. So we try to increase the signal difference between the capture probe and the target probe. As large as the signal difference, your sensitivity can be increased. This is the idea for tuning the surface chemistry using different molecules. So here, for example, here, this is the single-stranded DNA and this one is the double-stranded DNA. And these two are formed based on perfect helical structure. Whereas in the input graph, where we have NC means non-complementary, that means helical formation does not happen on the surface. Therefore, we did not see any change in the signal. Therefore, change in the signal. And here, when we have a single point mutation, the signal lies between these two. And here, this is the concentration variation of a target DNA. And we use, yes, sir? We use the signal difference between
white cement.
we use two different techniques. One is the cyclic voltammetry and another one is the impedance. What we do, we calculate the resistance between the single strand and the double strand difference, and we plot as a function of concentration. What we see was the complementary gave higher resistance than the non-complementary, and the lowest detection of the helical formation was detected at 100 attomolar. 100 attomolar. Similarly, here we again combined with the two different lipids like DOTAP and DOP mixed together. Again, the same protocol was used. Confirming the attachment of a lipid layer on the surface, and we put a single strand and a double strand. Single strand is this one, lower resistance, and a double strand is this one, higher resistance. And here the concentration linearity range we obtained is this is the femtomolar, femtomolar to the nanomolar range, and the lowest detection limit is 10⁻⁻14 molar. And similarly, recently what we did was we also used, this is the summarization of using of lipid along with gold nanopod. These works were developed in our laboratory and published in various journals like bioelectronics, biosensor, sensor actuators, analyst, and analytical chemistry, like that, for various diseases. And mostly we focus on the pathogenic disease diagnosis or cancer disease diagnosis. And here are the lowest detection ranges we obtained using different types of molecular layer used for diagnosing single-stranded DNA or disease, with the target DNA, which forms a double helical structure on the sensor surface. And recently we also replaced, because lipid layer, we get quite reproducible results most sometimes. In order to avoid that, we try to change again the matrix for attaching the strand using the metal oxide. Here we took the the tungsten oxide gold nanoparticle, and then we put it to the single strand and double strand. The again we also use a graphene core. All these things are compared here. The surface chemistry, when you change it in surface chemistry, the concentration of DNA sensing limit can also, at the linear range, are also being changed. This is what I would like to convey here. And this is the recent result, where direct use of the tungsten oxide give you the result of sorry, the previous slide summary is again by using the impedance spectroscopy. And similar way, we also try to develop the immuno based sensor. The principle is that when you put antibody on the surface and you have an antigen in the blood, and this antigen interacts to form a complex that will generate a signal, and you can use the enzyme label or without label also you can monitor change in the current signal before and after interaction of the target molecule like antigen. Here we use the streptavidin biotin interaction is also there, which is all these enzymes and streptavidin biotin to enhance the signal intensity. This based on this method, we also published a paper in Electrochemical Acta, and other one more journal is Sensors and Actuators. And this one, and here this one is the H1N1 detection, viral detection, or we call it tuberculosis, sorry, tuberculosis, TB. This one is H1N1 viral particle detection using immunosensing methodology, where we put antibody on the surface and we have antigen from your viral particle, which is from the patient. So we put the antibody and we have antigen from the patient sample. These two interact, which will generate the signal. Here, this one was not from our laboratory. This is from the other laboratory, where they developed a colorimetric sensor. When this complex formed, it generates a color. Depending on the intensity of the color, you can quantify the concentration of the viral particle in your blood sample. This is another diagnostic one. This one is similar to the pregnancy test, where you get the formation of colored line indicates the pregnancy status, and here the same principle can be extended for developing immuno sensor kit, where it is applied for indicator labeled. This one is used for COVID test kit, the COVID diagnosis, where if you get the colored line, you will be diagnosed as affected by COVID. If you don't get a color, this kit is already in the market, like already in the medical shop. It is cost about 250 to 300 rupees, where you put your blood sample, it indicates the color formation. If you get a color formation, that is you will be affected by COVID virus. So like that, the sensor devices can be developed based on the molecular sensing. However, my conclusion is that innovation in a sensor design and development is critical for smart sensing of diseases at early stage. Early stage disease sensing is required to control the mortality rate. I think I conclude my lecture at this stage, and I thank my all fundings from different funding agencies of Indian government, and also from Alagappa University, and my scholars who are working on these projects, and they were awarded 7 PhDs and 2 are undergoing, and some master degree students are also involved in this sensor development in my laboratory. I thank you, the organizers, for inviting me for this faculty development program through Atal Cell. I thank you once again for the given opportunity. I like to take any questions or clarifications if any. Thank you, sir. Thank you very much for your patient listening. Sir, any queries or suggestions or clarifications? Hello? Am I audible? Is anybody there?
Yes, sir. Audible, sir.
Yeah, any queries, sir?
No, sir. No from my side.
Or any
Really,
suggestions?
I'm very much happy to
Yeah,
listen
sir,
to
you are audible.
Yes. Am I?
Yes.
I hope I spoke a little bit faster to cover all the diagnostic methods we developed in our laboratory, little bit with the basics. I spent most of the time with the glucometers than on the other sensor development. So if you give another 1 hour, I go with the basics. If there is no questions, I thank once again all the participants for your patient listening and the organizers of this faculty development program through ATAL itself for your invitation and the opportunity given to me. Thank you once again. Thank you one and all.
Yeah, sir. Thank you, sir. Once again, we will ask the participants any queries and then we will go for the formal vote of thanks.
Yes.
Participants, if you have any queries, please raise your hands or put in the chat box. Or if not possible today also, please mail us. We will ask the sir personally also, and then we will send the queries answers. So if now itself means please raise your
Most
hands.
welcome, madam. You can send any queries or any clarification through mail also. No problem.
Thank you for that, sir.
Yes.
Any queries, participants? I think no, sir. Shall we proceed the vote of thanks, sir?
Yeah, sure.
Yeah, sir. One Vijay Raj sir is asking, sir, please.
He is my student, madam.
Okay. Then shall we proceed the vote of thanks, sir?
Yes, sure.
Yes, sir. So on the behalf of Siddharth Institute of Engineering and Technology, ATAL FDP, my deepest gratitude to you, sir. First of all, thank you for your time gracing us, and then your valuable time, because you are having busy schedule. In that evening also you are giving the time. That is very we will gratitude for that always, sir. And then your session is start with that molecular sensing of disease diagnosis, and then about the molecular sensing, this and all the vast topic, but you clearly draft that, sir. Thank you for that. And then this is motivate us also to do the more research about this. So I hope all the participants are feeling the same. In the future also we need your collaboration, sir, for any this like FDPs and all. So thank you for your time, sir. Thank you so much.
Definitely, madam. I will always available for your program. No problem. Thank you.
Thank you so much, sir.
Thank you once again. Shall I close, madam? Then?
Yes, sir.
Thank you.
Thank you so much for your cooperation, sir. Thank you, sir.
Yes, sir.
Participants, please stay back, because we will post the Google link. So please fill the Google link attendance. It will be considered as your attendance for the FDP. So participants, please stay on the line for few more minutes. We will post you the Google link attendance within few minutes.
Do you have another lecture, madam, following this?
No, sir. This is the end of the session, sir.
Okay.
Dear participants, one small information. Now I posted the Google link attendance. So please fill the attendance. It is mandatory. It will be considered as your attendance. Please post your attendance in the Google link. I hope everyone saw the Google link in your chat box. So please fill the link. Ma'am, Priya ma'am, I'm audible.
Hey, abang pakala. Kaya बैठ तो तू बैठ जा. காந்தி का प्रश्नाला अब हमें तमिलनाडु ठीक बोलला हम फॉर्म चकले थी एक उन आ काय है बा हम आल्सो डेन को फॉर्म डिस्प्ले थी नन प्लीज ने नस्कान निकाल के अंदर सही है बा बस आप कर नहीं कर